Unfortunately, these criteria aren't taught so much in school, but I learned them through 28 years of being a scientist.
This week, we are excited to welcome Dr. Robert Stadler to the show. Dr. Stadler is a true expert in engineering, biology, and medicine, and epistemology. And Doug, I mean an expert from back when an expert was someone you actually could trust.
Oh, yes. I remember those days, Fred. And researching Dr. Stadler, I was reminded of why experts are good and they're very necessary, especially experts who are not afraid to be challenged with real hard questions. Dr. Stadler is the author of The Scientific Approach to Evolution, What They Didn't Teach You in Biology. He got his PhD in medical engineering from the Harvard MIT Division of Health Sciences and Technology. He's a scientist in the medical device industry, Fred, where he's made things that actually work for over 2 decades. He's contributed to cardiac devices implanted in millions of people all over the world. He's been elected a fellow of the American Institute of Medical and Biomedical Engineers. His 20-plus articles and papers appear in a host of peer-reviewed journals, and he's approaching 200 U.S. patents. Dr. Stadler, welcome to Real Science Radio.
Thank you very much, Doug. Great to be here. I appreciate the opportunity.
Amen.
Yes, Dr. Stadler, you've got a presentation for us, and you're going to tell us about your book. So would you like to go ahead and dive into that? But before you do that, Dr. Stadler, I got to ask you, what's your favorite invention that you've made for the medical industry? Is there one that stands out?
My favorite invention. I focus on heart failure, and people with heart failure, when they get into trouble with heart failure, they retain fluid. They kind of blow up with fluid in their body, and so we have a device that measures the impedance across the chest, and the impedance is inversely related to how much fluid is in your body. So we can track the heart failure status by this measurement every day and tell us when people are getting into trouble or not.
Wow.
Okay, that sounds pretty cool. Now, since you said the word impedance, I have to ask, is there some kind of electrical field involved? Or is that a different kind of impedance? You're talking to an electrical engineer. So
Yeah, I always say it's hard to resist a good explanation of impedance, Yeah, you're putting a little electrical field across the chest that you can't feel. It's very small, but it does the job.
Okay.
Wow.
Cool. All right. Okay, so let's go ahead and dive into your book, The Scientific Approach to Evolution by Dr. Rob Stadler.
Yeah.
What They Didn't Teach You in Biology.
Well, thank you. So I gave this the quick, easy title, which is Evolution Good Science? But because we're here on Real Science Radio, we could also say, is evolution real science for your show? But I'm going to start off with a little bit of a dramatic beginning here, that we are currently in the midst of what we call the information age. And each day there's this deluge of information coming at you in all kinds of directions. And each of us is surviving the information age by filtering out, in other words, focusing on some forms of information that you trust in, and then rejecting or pushing aside other forms of information that you don't have the time for, or you don't trust, you don't like. So surviving this deluge is all about this filter we have, and the filter we're developing over time as to what information you focus on. You've probably heard the old saying, you are what you eat. And in the information age, I think you are the information that you digest, right? Because it kind of defines who you are. And today we're going to talk about how to decide what is trustworthy information and what isn't, in the focus on the field of science. What kind of science gives you confidence and you should trust it, and what kind of science is really not giving confidence and you shouldn't trust it. And in doing that, I want to zoom in on the topic, everybody's favorite topic here, which is evolution. And when I mention evolution here, I'm talking about the big scale evolution. where a single cell over billions of years has become all of life that we know today. Everybody's familiar with that. We're all taught it in school. But that is very much in contrast to what the Bible teaches, which is more of a forest of life, or some people call it an orchard of life, where you have many different trees, not one tree, and each tree represents a kind of life. And Genesis 1 mentions the word kind like 10 times. So it's very important, very emphasized here that creatures reproduce according to their kind and so forth. So we have the very familiar dog kind, where a wolf over time has produced the whole diversity of dog breeds that we know today. But they're all still dogs. They're all still part of that tree. This is way oversimplified, of course. It's kind of a cartoon, but it gives you the idea. So to just dip our toe into this, and we think of what is science telling us as far as which of these two is correct. That's what this is all about. What does science say about these things? Well, science fundamentally is about making observations. And in order to make an observation, a human has to be there and to make observations and learn about how natural processes work, how do natural laws work. And they have to be able to pass that down to the next generation so we all learn from it and understand how natural laws work. But on this tree of life, humans here only existed for a small amount of this tree. And before that, there really is no way to make any direct observations because humans weren't there. That doesn't mean that science has no place going back in time here. It just means that it becomes more tenuous. It gets cloudy and foggy back here, and it's pretty hard to discern what actually is going on. We have to admit that, really. Now, if you do this and look at what part we can observe, humans have observed, the tree of life and the forest of life are basically identical for what we can observe and have observed. And I think that's very telling. So what we are here to look at agrees with what the Bible says is the key. But we're going to go a little deeper into the science now, and that was kind of a cursory overview. We'll go a little deeper. So according to science, which of these is correct? That's really the question we have in mind. And if you're a good scientist, you keep an open mind and you consider both possibilities. You accumulate evidence through observation, through experiment, and you decide which of these two hypotheses is better supported by the evidence. That's how science should work. So that's what we're going to do. Now, most scientists up front, of course, they'll say that the evidence for evolution, the tree of life, is just overwhelming. And you'd be a fool if you would disagree with that. That's what they would say. The evidence is all stacked up in favor of the tree of life. But there's really two problems with that. And the first problem is that up front, they block out the other possibility. They say this isn't science, it's pseudoscience, and science can only address natural processes. And so we have to only consider evidence of natural processes. So your biology textbook basically is kind of like a sales pitch, a sales brochure. It only talks about one side and is very positive about the evidence for that one side. And I think we're all aware of that. But this is an incredible form of bias in science. If you're so biased that you exclude another possibility up front, that's really bad bias. That's the worst bias and a sign of bad science. And the second problem we face, this is more subtle, and this is what my book is about, is that in doing this, they're actually prioritizing weak evidence, or what I call low confidence evidence. And they're prioritizing that over stronger evidence or higher confidence evidence in order to make this claim. And this is what we're going to focus on, because I know this is kind of subtle, and we're going to try to make sense of this statement here. And that's what my book is about. So I'm going to focus on my particular area of science just as an introduction to this idea. And that would be medicine. And medicine, I think we can all agree, is very important. I would argue it's the most important application of science because it's life and death. It determines if you're going to live or not. And when you take a pill or you go through some procedure, you really want good science behind what you're doing there in medicine. Doesn't mean it's perfect. It doesn't mean there's no errors, but it's important, and society has put a lot of pressure on it. And because of that, or as a result of that, doctors have gotten together and agreed on a way of prioritizing evidence, medical evidence. And we don't need to look at the details on this table. The point is, I just want to show you that doctors have agreed that evidence up here is given high priority. This is credible, high confidence kind of evidence, where evidence down at the bottom is low confidence evidence that's not prioritized. So in other words, if I invent some new pill and I claim that this is going to cure cancer, right? I have to go to the Food and Drug Administration, the FDA, and try to convince them that my pill can cure cancer. Well, the FDA is going to be looking for evidence up here at the top of the chart. And if I only show up with evidence at the bottom or near the bottom, they're going to kind of laugh it off and push me aside. That's how this works. And notice at the bottom is consensus of expert opinion. So if I bring in a few experts and they say, I think this pill is going to cure cancer, that's not considered good evidence, right? That's not going to solve, that's not going to get me through the FDA and get my drug approved.
Yeah.
So when I bring this up, some people get all twisted up thinking, oh, my criteria here only apply to medicine. Well, that's not true. That's not my point. I'm just using this as an introduction. My point is that all forms of science should be able to prioritize the best evidence on top of lower quality, lower confidence evidence. Everybody should be able to do this, and I applaud medicine because they've done it. So how do we broaden this out then to apply to all kinds of science? And that's what the book is about. And here I am proposing 6 criteria. These 6 criteria helped you to separate confident evidence versus low confidence evidence. And I don't mean to make it sound like it's one or the other. It's actually a spectrum. You can have high, medium, medium low, low confidence, depending on how you address these 6 criteria. And I'm bringing this up also, going back to that filter of the information age, that I hope everybody can get familiar with these 6 criteria. And then tomorrow when you hear news, some new science discovery, you can apply these 6 criteria to say, how much do I trust? How much confidence can I place in what I just heard there?
Well, Dr. Stadler, first of all, I'm sure you realize that there's a significant segment of the population that thinks you're awfully presumptuous to assert that the average person should be allowed to develop their own filter to judge what's right and wrong. Simultaneously, though, each one of them, in choosing a medical procedure or a medicine for themselves, would agree with everything you just said.
I totally agree with that, yes.
Fascinating.
Yeah, and I do hope that everybody develops their own filter. My filter's become really acute, I think, so that when I listen to the news in the morning, often a little red flag pops up because it doesn't meet these criteria. And I'm like, I don't trust what they're saying,
Yeah.
So I hope people can use that and benefit from it. That's the point of this. So you might be asking, what are these 6 criteria? So let's get into that. The first one should be super obvious to everybody, that evidence that is repeatable gives you higher confidence, right? So if, I like the simple example of, let's use science to find out how fast an object falls when I drop it, right? I just take an object and drop it and I measure how fast it falls. And I can do that again and again and again. And every time I repeat it, my confidence goes up. And if someone else repeats my result, the confidence goes up. Someone in a different country repeats my result, right? It's obvious. So things need to be repeatable in order to give you more confidence. It doesn't mean that if you can't repeat it, there's absolutely no value to it. It just means you admit that this is a bit tenuous as a conclusion, right? I don't have total confidence in this. Number 2 is things that are directly measurable give you higher confidence than things that are indirectly measurable. And that's a little more subtle. Let me give you an example that when I was a kid and I felt like I was sick, my mother would try to measure my temperature, and the indirect method she would apply was to put her hand on my forehead and say, I think you have a fever. It's indirect because it's measured through her hand and her subjective decision process, and her hand might be cold or hot when she does this. It's not very accurate. I think you get the idea. The more direct way, of course, is to measure my temperature with an instrument directly to the source at the core of my body, right? That's how you do it directly. Another example of this might be like if you want to study a black hole out in outer space. We all know that black holes don't actually emit light and everything, they're sucking everything into themselves. So you can't make direct measurements on a black hole. It all has to be very indirect, and that gives you lower confidence. If you could bring a black hole into your laboratory and you could poke it and prod it and measure stuff from it, then you'd have much more confidence. But we can't do that. It's just a fact. Number 3, you have more confidence if your study is conducted prospectively. And that means that you plan ahead how you're going to do the study, and then you conduct the study according to your plan. So why is that so much better than just a retrospective study where you look back into the past? Well, with a prospective study, you can develop your plan in a way that blocks out anything that's confounding, anything that's going to confuse or get in the way. You can plan ahead to block that out and to get at the truth, right? So, for example, if I'm dropping an object and seeing how fast it falls, I can plan ahead that I want all the air out of the room, because air inside the room, or maybe wind or something in my room, is going to interfere with my measurement. And that's a problem. So I want to plan ahead to get that out of the way. That's prospective. Okay, number 4 is avoiding bias. And I think everybody can agree with this, that we all have bias. I have bias. Everybody has bias. And it's kind of like glasses that you wear, and you're looking through those glasses through your bias and perceiving everything according to your bias. So in science, bias is just devastating, and you can't just say, I'm trying not to be biased. You got to work at it. You got to have some techniques in mind that pushes bias out of the picture, or else it's going to corrupt what you're doing. Next one is avoiding assumptions. Assumptions in science are kind of like taking shortcuts. They're often used to save money or time, or to fill in something that you really don't know. And that's really dangerous, right? Because you're making assumptions, gets you into trouble. So in my example of dropping an object and seeing how fast it falls, I mentioned that the air in the room is kind of interfering. But getting the air out of the room is going to cost like $1 million, and I have to take 6 months to do it. It's going to delay the project. So can't we just assume that it's not a big deal to have air in the room? So this is the kind of thing that happens all the time in science. It's unavoidable to get rid of all assumptions, but the assumptions you make, you should openly disclose, freely talk about them, and admit that they might be causing some trouble. All right, all that makes sense so far? You guys good?
Yeah.
Oh, this is very valuable stuff. This is great.
It'll be interesting to see how you apply this to the theory of evolution.
Yeah. Yep, that's coming.
Okay.
All right, I got one more. Number 6, the final one, is about making reasonable claims. And this is more about when the experiment is done, or when your study is done, and you have to write it up and say, here's what we learned. You got to make reasonable claims. I like an analogy here of don't get out over your skis, right? You got to stay on top of your skis, which means only conclude stuff within the area that you studied and not extrapolating off to some other area. For example, if I measure how fast an object falls here in this room, I shouldn't be assuming my conclusion applies to the top of Mount Everest, or if I was on the moon, because that's not true. It's not the same force of gravity up on Mount Everest or on the moon as we have here. So that would be an unreasonable claim if I were to do that. And I like to use this can of nuts also as an example because these nuts, they want you to buy more, of course. And so they say they're heart healthy and they're approved by the American Heart Association. But the FDA always has to step in and say, well, what are you claiming here about how healthy these are? And they want to make sure that you're accurate in your claim. So on the can of nuts, they were required to write this statement shown here that scientific evidence suggests but does not prove that eating nuts may reduce the risk of heart disease, right?
Yeah.
And I appreciate how honest this is. It has these hedging terms, suggesting and may, and that's what the real evidence says. So they did a good job making reasonable claims. I appreciate that.
Got it.
So the next time you eat nuts, make sure you check the label, right?
Well, and Dr. Stadler, I just want to thank you because these 6 methods or mechanisms that you've introduced, these are valuable for assessing pretty much everything that we get through the avalanche of information that you described earlier, because we often see clickbait headlines that appeal to our pre-existing biases, and then we click through and we read, and it's good to have this guidance to help us to avoid going down rabbit holes that are attractive to us, and besides just being deceived with lies, but also just chasing foolishness. This is very good stuff.
And clickbait is all about breaking this rule here of making reasonable claims because they want to amplify it. They amplify everything to call your attention. And amplification is a big problem when it comes to claims, right? If I have a 0.1% improvement, I can't say, great results, best ever result. That's what you're going to see in the clickbait. So I want to contrast that with this statement right here came out of my son's biology textbook shown here. So this is a direct quote. For example, organisms as dissimilar as humans and bacteria share genes inherited from a very distant common ancestor. So you can see in this statement there is no hedging language like you have over here. I think it would be a much better claim if they said, we think, or suggest, some evidence suggests that this is true or something like that. But no. They're saying with absolute confidence, they're telling you that we have genes shared from a very distant common ancestor.
Yeah.
Now, I don't think anybody debates that humans and bacteria have some shared genes, because that we can directly observe, we can measure it again and again. Nobody doubts that. The part that you doubt is, were they inherited from a very distant common ancestor? That's the part that you can't observe, you can't repeat. And making a strong claim about that is disturbing to me. So, looking at these two statements and the contrast between them, you're kind of I'm left scratching my head, wondering why is our society more concerned about claims that we make about nuts than claims that we make about evolution? I mean, which is more important? Okay, so let's step back again. So I want to show you an example then we were talking about this example of how fast does an object fall. I want to answer this question. And that can be done while meeting all 6 of these criteria. If you do a good job, you'll meet all 6, and you can just knock this out of the park, right? Science can give you great confidence in answering that question. I think we can agree. So to contrast that, we can take this statement out of my son's biology textbook. What this statement is doing, it's answering this question: do humans and bacteria share genes from a common ancestor, right? That's a question you can ask of science, and you hope science will answer it for you. But the problem is, trying to answer this question, you can't meet any of these 6 criteria because it can't be repeated. You can't go into a laboratory with some single-celled organism and then watch it evolve into bacteria, watch it evolve into humans, and show that the genes actually have been inherited all the way down their line, and you have common ancestry. You can't even come close to repeating it. It's just the opposite. Nobody witnessed the actual event. That's the opposite of directly observable. There's nothing directly observable about sharing genes from a common ancestor over such, billion years. It's extraordinarily retrospective, looking back into a billion-year past. You can't get much more retrospective than that. That's the opposite of the prospective kind of study we like. Does anybody think that there's any bias involved here? The people who write our biology textbooks and so forth, do you think they have any bias? Like I said, we all have bias, but I think it's really standing out here. It's rather blatant and obvious. What about assumptions? Honestly, I think this entire statement is built on an assumption. They're assuming that if I see a gene in bacteria and a gene in humans that are pretty much the same gene, then I'm assuming that must have come from common ancestry. Another option would be a common designer. A designer put this gene in bacteria, a designer put the same gene in a human, but that gets excluded up front. We're assuming that away. That can't exist. So basically, you're assuming it's common ancestry. The whole thing is built on assumption. And then finally, overstated claims. We already went through that with this not having any hedging terms and being built upon assumptions. So this statement really belongs way over here in the very low confidence bin. And when you see statements like this tomorrow in the news or so forth, you should have a little red flag going up in your head that it's not giving you confidence because it doesn't meet any of these criteria. Does that all make sense?
Yeah, definitely. I mean, I think number 5 too leads to, isn't that kind of circular reasoning too? They assume as naturalists, they want, naturalism to be true because they have a bias against God that they assume it's true. So then they make what you point out here is an assumption, and as if the assumption proves naturalism. And it's just circular.
Yeah, and it's very important to disclose your assumptions. It's when assumptions are hidden, that's when you really trick people, So you always have to question what assumptions people are making. And if you get those out on the table, then it becomes more clear, right, of what's going on here.
Yeah, so are there disclaimers at the front of the 9th grade biology book that declare the assumptions and the biases?
Not that I know of.
Well, there wasn't in mine. I remember that.
Right. It would be nice if they were a little more open, a little more candid about that. I would appreciate that very much.
So I wonder if it's wrong to assume, since the biases and assumptions are not declared, when I would assume, doctor, that most of the people who write these books are familiar with the concepts that you're taking us through here. They're scientists, some of them.
Yeah.
Right.
I think, unfortunately, these criteria aren't taught so much in school, but I learned them through 28 years of being a scientist.
Oh.
I wish they were taught in school.
So are you saying that, forgive me because I'm not a scientist, but I would have assumed that everyone who's had a scientific education would be familiar with this, and you're saying that's not necessary. I shouldn't make that assumption.
I would say these are not directly taught, but they're kind of inferred. Like everyone knows repeatable is a good thing, right? Everybody, and prospective study, people get the concept. But here's where the problem lies. If you decide to go into a field like paleontology or anthropology, right? You go into that because you're interested in it. You have a passion for that, and that's fine. But when you go into it, you're not told up front that this is a very difficult area to obtain truth because we're looking back into the past and it can't be repeated, it can't be directly observed, and we can't do prospective study. So you're going to spend your career somewhat frustrated trying to get to the truth when it's all very cloudy and murky. Nobody tells them that as they enter into that field. And the people that are experts in that field are probably rather offended by what I'm suggesting here, because they think their field is just as confident as anybody else's. And it's just simply a fact that it isn't, and it can't be as confident as some prospective kind of fields you can work on.
Yeah, that's a great point. So I'm going to bring up something I know isn't, it's going to offend some people. It's not, but when I look at your slide here and you think of the different scientific disciplines, uh, I know what you're talking about when you go into certain fields. And again, I've got friends who work in I've got friends who are geologists and geophysicists. And to me, those kind of push over to the low confidence field because you have to, you look at your 6 criteria here and you fall into those a lot. And you have to work really hard to try to, find useful information. And you can improve your odds, for example, when you look at geophysics and trying to find oil and natural gas underneath the earth, but you're dealing with a lower confidence, and it's just it's a challenge. But what I've found is sometimes it's presented as if it's high confidence, and it just isn't.
Right.
And
Thank you. That's
that's
right
what.
on. And I do want to emphasize that in no way am I calling these folks bad scientists, right? They're honestly trying to find truth. They're working hard at it, and they're using the best available tools and techniques. It's just that in the end, what they come up with can't have as much confidence. And it doesn't make them bad people or bad scientists. It's just it'd be nice to admit up front that you can't achieve the same level of confidence. It's just a fact.
Yeah.
Yeah. And I did want to ask you also, looking at your 6 criteria. So I worked for Criterion BCT, and I love your analogy of, so I 1 year in the medical industry as an engineer. We did a chamber that grew adult stem cells, but they had a blood circulation device, and the team to test that was enormous. So we're not just talking the engineers who developed it, the test team to design tests around it. And you talk about, okay, someone's a lot more interested in the eating healthy nuts than they are about evolution assumptions. People are, when it comes to life and death, and people are super interested in the high confidence area of your chart here. And can you speak to, I know you work in the medical industry, the medical technology. Testing is probably is a big part of this, and I think maybe that falls under repeatable and maybe directly observable. The first 3, really, if
Yes.
not even all 6 of them. So that's another thing that with the low confidence, can you even test it?
Yeah, and
We spend a lot of money on test engineers
yeah,
in
it is
technology.
predominantly where the money in development goes is testing stuff and repeatedly testing stuff and proving that it works. It takes an enormous amount of resource. It's just built into the business. You have to do that. And yet, in my 28 years, it's still, even though you go through all that, there are still times when you are surprised and humbled by things not going the way you thought they were going to go. Science is really hard. And often
Mhm.
when things don't go the way you expected, you look back and you figure out there was some hidden assumption that was made. And that's what came back to bite you.
Yeah,
Yeah.
some corner case you just didn't think of,
Yep.
or what? Yeah.
That's what
Yep.
happens. So now let's go back to the big topic in mind. So we have these 2 hypotheses, and we want science to tell us which one is preferred. And I say preferred because, more evidence could appear later that might change your mind. You got to be open to that. So we're going to stack up evidence and see which one of these is preferred. So let's look at this tree of life and see what kind of evidence we have for large scale evolution here. What kind of evidence do we have? Well, if you look in your biology textbook, any biology textbook, or even lectures on this or in the media, basically you'll see 4 kinds of evidence presented very commonly. And I'm sure you're all familiar with the fossil record. They say that very much supports this. Geographic distribution is about how different kinds of life forms appear in different locations on the Earth. And they say that supports this. Vestigial organs like your appendix. And then comparison between different life forms. This could be comparing like the bone structure of your arm compared to that of a dog or a bat or a lizard. Or it could be comparing the genetics, the DNA comparison, the molecular biology comparison, showing similarity between, your DNA and another organism's DNA. So this is the kind of evidence they put forward, and this is what is in the book to teach you, to convince you that this is how life came about, right? So I just want to emphasize this by calling on a friend here. We're going to ask Richard Dawkins the same question, and we'll show you what he answers about this.
Out of all the evidence used to support the theory of evolution, what would you say is the strongest, most irrefutable single piece of evidence in support of the theory?
There's an enormous amount of evidence from all sorts of places, and it's hard to pick one strand which is more important than any other. There's fossils, there's the evidence from geographical distribution, there's the evidence from vestigial organs. I think, to me, perhaps the most compelling evidence is comparative evidence from modern animals, chemical comparative evidence, genetic, molecular evidence.
Okay, so there you heard it right from Richard Dawkins, that out of these four types of evidence, the most irrefutable, the most convincing evidence comes from comparison of life forms. So taking one life form and comparing its DNA or its molecular structure or its bones or anything morphology-wise to other kinds of life forms. The problem is that this statement, we just dissected this statement, right? And we found that it met none of these 6 criteria for evidence. And what Richard Dawkins says is the best evidence that there is, the most compelling, right? The most irrefutable evidence is this kind of evidence. It is this evidence that has none of the criteria for high confidence, and that's the best that they got.
Right. And that's probably why it took him almost 30 seconds to answer a very direct question. So I've had to present evidence for things that I believe before. And when someone asks me, what's your best evidence? I immediately grab my best evidence and say, here, this is my best evidence. But he said, well, there's an enormous amount of avalanche of this, so much, it's hard for me to decide. And then he comes to what you've now revealed as the lowest confidence type of evidence. Interesting.
Yeah. And the problem is that all of these other forms of evidence also meet none of these 6 criteria. And that's, my book goes into it in detail. I don't want to take an hour to go through this, but like for the fossil record, it is repeatable to say, did a T-Rex exist? Okay, I can answer that repeatably because I can go dig it up again and again and find T-Rex. But if you're going to tell me, did a T-Rex or did dinosaurs evolve into birds, that I can't repeat. I can't make that happen again in the laboratory. I can't directly observe that happening. I can't prospectively study that. So trying to answer the important questions of where life came from with this data, you're going to fail all 6 of these criteria. And that's the best we got.
Yeah,
So,
vestigial organs, that one's just flat out wrong. I mean, isn't there time? I mean, you're in the medical industry, you know, time and time again, they're disproving these things that they claimed were vestigial, like the appendix, like tonsils.
yes, and junk DNA goes into that bucket also. Right?
Oh, true. Yeah.
So all of this stuff is very, not just low confidence, but it's really down there at the bottom of the bin. And I need people to appreciate that. So you're left with the question, okay, is there any high confidence evidence that speaks to evolution, right? Because that's what we should be focusing on. That's what we should be prioritizing. And there's a bunch of experimental evolution studies that can speak to this. So we have Richard Lenski, who studied 80,000 generations of E. coli in his laboratory. You're probably familiar with the study he did. 80,000 generations of E. coli, if you turn that into human years, 80,000 generations of humans, right? You're talking about a couple of million years. And what's the best thing he found through this study? The most innovative change he found in this bacteria was after 33,000 generations, the E. coli were now able to metabolize or to eat citrate. And so interestingly, the E. coli always could metabolize citrate. They just couldn't bring it in. They couldn't transport it across the membrane. That was their problem. But also interestingly, E. coli always can transport citrate across the membrane if there's no oxygen present. But his study was performed with oxygen present, and so his E. coli couldn't get citrate across the membrane, and that was the problem. So this is from Richard Dawkins. It's a quote from his book, The Greatest Show on Earth. And you can read that and you'll see that he referred to this study and wrote a whole chapter on this study. But he called it a beautiful demonstration of evolution in action, new information entering genomes and combinations of new genes. So keep the focus here on the words new, because later on, after he published his book, they actually studied the genomes and studied what had changed in the genomes. And in these two papers, basically they showed that the ability to metabolize citrate came out to be duplication of one existing gene. And we know that duplication of a gene is a very common kind of change that happens, much more commonly than like a single point mutation. So this is very common, and it involved enhanced expression or enhanced use of another existing gene. So it's two mutations that occurred to existing machinery, nothing new, no new genes, no new information. So I think what Richard Dawkins concluded was an assumption from his bias up front before we actually knew, and he really kind of blew it on that one.
Yes, yes. Now, Dr. Stadler, I have to stop you because you just used the word mutation, and I wonder if, is it really appropriate to call what happened with the E. coli colony, is it really appropriate to call that a mutation? Wouldn't it be more appropriate to call that an adaptation or an expression?
I thanks for bringing that up. And I don't think we have the exact answer, but I do think that it could be any of those possibilities, right? It could be a directed mutation, meaning, the organism chose to do that itself. It could be a change that is preferred, not exactly random, not exactly drawing from a lottery ticket, but something that's more likely to happen, something that's preferred. And that's what I believe it was. But it's really hard to tease apart true randomness from something less random or even directed.
Right. So and so just as a layman, when I hear the word mutation, I think of Chernobyl and a wolf that develops extra legs, and it's not good. A mutation generally, I just assume it to be harmful.
And generally they are. I mean, probably 95+% are going to be, well, a large percent are going to be harmful. Some will be neutral, and then very, very unlikely few that could be considered beneficial under certain circumstances, right?
Spider-Man, right? The ability to shoot webs.
That was a really good mutation, yeah. Okay, let me move on. So there's other evidence like that. And I got to emphasize that Richard Lenski's experiment, if done properly, meets all 6 of those criteria. It could be repeated. It was prospectively studied. It was directly observed. They looked at the DNA, looked for changes. You got all that. You could accuse him of bias, maybe. I think he was kind of biased. Anyway, this is another study, one of my favorite studies. You can see the reference down here. And this one also meets all 6 of those criteria. So E. coli have the ability to produce tryptophan, which is an amino acid shown over here. And to build that necessary amino acid, they have this enzyme called tryptophan synthase. So these guys are really smart. They did a study where they intentionally damaged the tryptophan synthase and then see if the E. coli can evolve themselves out of this trouble because they need it. I thought that was very interesting. So what they did was they changed one nucleotide, one letter of the DNA, and for the gene that produces this tryptophan synthase. And they found that the enzyme now functions somewhat, but it's kind of poorly functioning. And then they put those mutated E. coli into a bath that had a limited amount of tryptophan. So there's a selective benefit if you can produce your own tryptophan, right? And what they found after about 100 million E. coli were grown in the lab this mutation was repaired. And maybe that was random, or maybe some direction was going on, but the E. coli figured their way out of it. And the E. coli with the repaired gene took over the population because they could make their own tryptophan. So you could give kudos to microevolution for having succeeded here in changing one nucleotide. So then they changed a different nucleotide. They tried it again, start over, fresh, with just one letter of DNA changed. And this one was more devastating. The enzyme now had no function at all. And they did the same thing, putting it into a solution with limited tryptophan. And after about 100 million E. coli were grown, again, the mutation was repaired. And the E. coli could produce tryptophan and take over the population with the better version of E. coli. Okay? Now, this is where it gets interesting, because now they're kind of testing the fringe. They're testing the limit of what microevolution can accomplish. What can it accomplish here? So now they make both DNA mutations. So there's 2 letters of DNA that are wrong in this enzyme, or in the gene that produces the enzyme. And now the enzyme has no function. And now they grew over 9,300 generations of E. coli in the same solution. And this is a huge amount of organisms, and it takes a lot of time. And they found that not only did the E. coli not get back to producing tryptophan, but the genes that make the tryptophan were either deleted, like thrown out as garbage, or just suppressed so that they're not producing an enzyme at all anymore. And that's because producing this enzyme, this protein enzyme, requires tryptophan. It actually is using tryptophan to make this thing, and it's wasting it's kind of wasting limited resources, and it just says, I give up. I'm not going to do this anymore. And that was the happy place for this E. coli to be. So if we kind of summarize what happened here, because I know there was a lot of details there, but we're talking about evolution not being able to repair an enzyme that is already 99.9% in final form. Okay, we're talking about an enzyme that takes about 2,000 nucleotides of DNA to specify the production of this enzyme. And all they changed was 2 letters out of 2,000. So that's 99.9% perfect. All evolution had to do was push it over the edge. And it not only failed, but it actually went the other way and basically jettisoned the whole project. So how could this happen? Doesn't this say a lot to us that it can't evolve from 99.9% of the way? Talk about a head start. And this is high confidence again. It's fulfilling all the criteria for high confidence evidence. And this tells us very clearly that evolution has some serious constraints. And then I'm going to throw in 2 quickies here. We've been working on mice in a laboratory for about 95 years and mutating everything possible and changing everything we can. And to put it bluntly, mice just stubbornly want to remain mice. You either get a dead mouse or you get a mutated mouse, but it's still very much a mouse, and we can't seem to get them to change into anything more significant. Same for fruit flies and zebrafish and domestic animals like dogs. Okay, and then this is a really recent paper of this little microcrustacean, and they studied it for 10 years and looked at its DNA over the course of those 10 years. And they ended up saying that they had an average effect close to 0, meaning not much is changing here over time. So all of this is high confidence evidence meeting those criteria. This is evidence that we should prioritize over fossil evidence or common ancestry evidence, the stuff that Richard Dawkins is talking about. This is the evidence we should prioritize. And this is clearly showing us that evolution is a very constrained, very weak process. Okay? So what we're seeing here, we take an organism and over time we subject it to some selective pressure, right? You either figure out how to make tryptophan or you're going to die, right? And you keep doing that and you're trying to change the organism, trying to see how far it can go. And then you can look at the DNA directly and say, what's changed here between this and this? And we found that in Lenski's study, the greatest innovation they came up with was simply copying one existing gene and increasing the use of another existing gene. It's not what I would call innovation at all. And then in the study of this tryptophan synthase, it couldn't even repair two specific mutations. So if we know how limited evolution really is, what happens then if we apply what we've learned to this scenario? You know, is this scenario possible given what we know about how evolution works, how it's constrained? And I just want to wrap up with, this is a very recent study, came out in Nature just a couple of months ago, and it is the best ever so far sequencing of human and ape genomes in giving us the ability to compare them. And I know this has been a hot topic of debate. Everybody argues over how similar chimp and human genomes are. And a lot of times you hear this quote of 98.2% or 98.6% similarity. This paper makes it pretty clear, and you can look at the figures here. You can pull these up. It's an open access paper. But they found that 13.3% of the human genome has no analog in the chimp genome. And this is called gap divergence. So you take a piece of human DNA and you look amongst the chimp genome to find something very similar to it, or close enough to call it a match. And in 13.3% of the human genome, they couldn't find anything that's analogous. It's a gap divergence. And then among those sections that do have analogous parts, which would be the 86.7% anyway, 1.6% of the individual nucleotides there are different, right? And then this analysis does not include the sex chromosomes, X and Y, because those are even more divergent. But anyway, the point is 13% of the human genome is something like 400 million base pairs. You could picture something like 1,500 books on a shelf. That is the difference between human and chimp DNA. So where did that come from over, you know, the supposed evolution going on here? This is supposed to be about 6 million years, and we have to come up with over 400 million base pairs of difference when we found from these experiments that we have trouble fixing 2 nucleotides.
Yeah.
Amazing.
It's not 400 million, but 2. So high confidence evidence, I think, is giving us a clear picture that this just doesn't make sense. So when we come back to this scale, we stack up evidence, right? I think we're making it pretty clear that high confidence evidence very much discourages this hypothesis, right? It's not favoring this hypothesis. And I think only low confidence evidence in your biology textbook, and that's under a lot of bias, a lot of assumptions, is being used to support this. And so the low confidence evidence is being prioritized inappropriately over high confidence evidence and giving us a very misleading picture. And we shouldn't be so surprised about that because Romans 1 tells us this is all going to happen up front. He said, for since the creation of the world, God's invisible qualities, his eternal power and divine nature have been clearly seen, being understood from what has been made, so that men are without excuse. For although they knew God, they neither glorified him as God nor gave thanks to him, but their thinking became futile, and their foolish hearts were darkened. So hopefully that was clear, and hopefully you get that. And hopefully tomorrow when you hear some news, some new discovery, that you'll be able to apply those 6 criteria, your filter, and hold on to the stuff that you can trust in.
Yeah.
Absolutely. So, you know, Dr. Richard Buggs, he has a blog here. He mentioned that article years and how close that study aligned with something he, you know, when he did a comparison back in 2018, you know, comparing chimp and human DNA. And,
Yes.
you know, yeah, the differences are remarkable. We've talked a lot about how the Y chromosome is 30 to 45% different. And so it just shoots down the whole argument that how could we be related to chimps? Mathematically, it's impossible. It makes that old Haldane's dilemma even worse. And the number of mutations you would need just to, you know, the number of breeding couples you'd have to have. It's like in the orders of 40, you know, tens of thousands of breeding couples to even give evolution a starting chance to maybe make one small change.
And it's interesting because all of those changes then would have to be fixated into the species. In other words, every human has to have those changes fixated, meaning that those changes had to be so beneficial that one lucky person that started this would be dominating over all other humans to get those into the gene pool.
Mm-hmm. Yeah.
Yeah. And how would selection ever see the change? You know, it can only operate on individuals, not at the genetic level. So
That's right. Good point. Yeah.
Well, and I want to address assumptions really quickly, Doctor. Is it safe for me to assume, based on the fact that, let's say I look at my son's ninth grade biology textbook and I see there's obvious evidence of bias, there's obvious regular appeals to very low confidence evidence, would it be wrong for me to assume that this is clear evidence of deceit?
Yeah, I don't like to do psychoanalysis. I mean, I think people are biased and their bias is leading them in a certain direction. I would agree with that.
I see.
Whether you take it as intention of deceit, that's kind of a psychoanalysis thing, but it
Well,
is
so
concerning.
that's one of the things I say about. I love the fact that you closed with the quote from Romans 1, because the Bible, when you read the Bible's actually reading you. The Bible is the original psychoanalysis device, but it's deeper. It's a soul analysis. It's a spirit analysis, and Romans points it out. And I want to just quote from Mark chapter 7, verse 21. Out of the heart of men proceed evil thoughts, adulteries, fornications, murders, theft, covetousness, wickedness, deceit, lasciviousness, an evil eye, blasphemy, pride, foolishness. All these evil things come from within and defile the man. And so just a note to parents out there, here at Real Science Radio, we're very open about our biases. And I want you to assume, if you see such things in the textbooks that your children bring home, that it is reasonable and it is advisable for you to assume that there is someone trying to deceive your child, and it's your obligation as a parent to protect your children from deceit. And what the doctor has explained here is clear evidence of not just bias, but deceit. And so be on guard against deceit, whether it's in the news or whether it's in your education or your children's education. Be on guard against deceit.
Yeah, and Dr. Stadler, you also have a book that you co-authored with Dr. Change Tan. We had her on a couple weeks ago, The Stairway to Life, An Origin of Life Reality Check. Can you just really give us a brief rundown of that?
Yeah, it was a joy to write this with Change Tan. And so this is about getting from simple chemicals, simple molecules to the first life. You know, today's presentation, we started with a single cell life and seeing if that evolved to all life. This Stairway to Life book is the predecessor, right? It goes from simple chemicals floating around in a prebiotic soup, if you will, to get to the first life, and showing quite clearly that natural processes oppose what needs to be accomplished in order to get life started. And this is not like God of the gaps, you know, saying we don't know how it started, therefore God did it. This is saying very clearly we know how natural processes work, we know what they do, enough to say that everything needed to start life is going the opposite direction. It's opposed to what natural processes do. And if you don't like to read books, down at the bottom of the page here in the corner is these series of videos that are on YouTube for free. Of course, you can look them up, but they basically describe the components of this book in an animated kind of fun way.
Oh, great. Okay. Yeah.
Oh,
we'll
yeah.
link to
And
that.
Dr. Stadler, just thank you so much for clarifying the the whole God of the gaps accusation, what you just said is just so valuable. The next time you're accused as a creationist presenting logical, reasonable evidence as appealing to a God of the gaps, remember what Dr. Stadler just said. What they're denying is the observation of known natural processes. It's not the God of the gaps. Thank you very much for that. That's very helpful to give us confidence.
Amen. Thank you.
Yep. Hey, and before we go, we almost forgot, Doug. Should we do the interesting fact of the week?
Fred,
Ready, keyed up for you.
Fred, this is perfect radio, or I guess we're video now, but you want to keep the audience on the edge of their seat for as long as possible. This is one of the greatest teases in the history of all media, Fred. I'm on the edge of my seat. Dr. Stadler, you may not be familiar, but every week Fred quizzes me on what he already knows to be the interesting fact of the week, and then he judges me mercilessly as to whether or not I'm correct. Fred, let's do it.
All right, so here is the interesting fact of the week. Which amazing creature can not only regrow its limbs, but if you cut it into pieces, each piece can grow into a whole new animal?
Oh. Okay, so like the first half, I thought I got this.
Okay.
And then the second half actually resembles the theory of evolution because no matter how many times we chop it up, I'm gonna guess the salamander.
Oh, that's really not a bad guess, but Dr. Stadler, do you want to take a crack at it? I never buzz my guess.
Okay. Sometimes I need to be buzzed.
Yeah.
I can't remember the name of this nematode, but it's very planaria. That's it. Planaria? There we go.
Yeah.
All right.
Planarian flatworm.
All right. Dr. Stadler, that's it. You want to drop the mic and walk off camera right now?
Hey, before you go, though, I do have one question I did want to ask you. So, in your experience, has evolutionary theory ever contributed to a practical breakthrough in your field, in medical science?
That's a good question. I don't know of anything. I mean, people would say like you could look at evolution of COVID, yes, COVID changed over time. I don't know if that has I don't believe that holding a belief in macro evolution, right? Single cell to all of life. I'm certain that has never contributed to anything practical value. But small changes, like we've seen, very tiny changes that can happen over time, those we observe, those happen, and that's a part of antibiotics, antibiotic resistance, and how bacteria has changed over time. So there's some value to that, but I think there's nothing about that that contradicts what I believe, because it's all been observed, and I think small changes happen over time, and we need to be aware of that. We don't have fixation of species, so to speak, and value to that.
Yeah, if you look at the history of where the word microevolution came from, the evolutionists coined that term because they wanted to take small-scale adaptation that God programmed into, life, into all the different organisms. We agree with that. Everybody agrees with adaptation, and so they had to throw in the word evolution, microevolution. But it's not large-scale evolution. So micro, small-scale adaptation, yeah, sure, we learn a lot from that. But
yep.
Well, and just real quick, this is one of the reasons I brought up the difference in just nomenclature between mutations and adaptations. One of the things I've heard that's made me laugh over the course of the debate around gain-of-function research is I've heard gain-of-function researchers talk about mutating a virus. And they say, well, we go in and we mutate and it's like, well, you're not mutating anything. You're designing, you're engineering. To me, that just strikes me as a layman, is that's the opposite of mutating. Mutating would be inflicting some random damage on the virus and then hoping it did. But sounds like what they're doing is engineering.
It's engineering. It's intelligent design. Yep.
Right. But because they are educated from a from an evolutionary worldview, they use ridiculous language for what they're doing. It's kind of comical, and it's why I'm of the opinion that almost all of the so-called gain of function research is the massive bamboozling of governments out of billions of dollars doing things that are virtually impossible. But we'll find out if I'm right or wrong about that. It could just be that they sound foolish to me, and so I'm underestimating their potential for danger and harm. That could be too.
Yep.
Yep.
Okay, so if you want to get into the nuts and bolts of this discussion, Dr. Rob Stadler has the book, The Scientific Approach to Evolution. You can get this at Amazon. Mm-hmm. You can also get the book that he co-authored with Dr. Change Tan, who, again, we had on a few weeks ago. That's The Stairway to Life. So, and also you can you can search for Long Story Short, Origin of Life. Search for that on YouTube, and we'll provide a link to it. And you can go watch I guess, short videos, Dr. Stadler, that presents your topic that you talked about today.
Yeah, there's actually 9 of them there that are about 12 minutes each, going through different topics.
Great.
All right. Scientificevolution.com, is that right?
That's right. That's the website.
Scientificevolution.com. And we will link to all the books on the ICR website.
Cool.
Very good. Right. Thank you guys so much. I appreciate the time together.
So yeah, and Dr. Stadler, I loved your compare and contrast between low confidence and high confidence evidence. It was a great illustration and just exposes just how weak evolution is. I really appreciate that.
Thank you. I hope it catches on.
Yeah. All right. So for Dr. Rob Stadler and my co-host Doug McBurney, I'm Fred Williams of Real Science Radio. May God bless you.